To us, that we decided to make this ARI1 and ARI2 was quite easy because we are owner of the two products to combine both. Most of the approaches make with CAR19 and CAR-BCMA from different companies, and to combine both, it's a little complex. Even the concept is very clear. If you have good safety of the treatment, specifically in autoimmune diseases, but in general, for all the treatment with CAR19 and CAR-BCMA, the adverse effect can be important, but in general are manageable, and the safety in some way, it's an important issue, but can be managed. The combination of both can increase the efficacy of the concept, because with CAR19 you don't kill plasma cells, and with CAR-BCMA you don't kill the precursors of the B cells that produce antibody. So our combination for us is a possibility to make a more deep effect in the pathway of the B cells and the humoral response, and specifically to demonstrate efficacy, as it's a clear need in a lot of different patients with non-Hodgkin lymphoma with large B cells. We decided to make the safety and the first studies of efficacies in this target, because it's clear that there are patients that lost the antigen CD19, that it's the CAR has been approved for this non-Hodgkin lymphoma. It's in most of the cases CD19. So if you lost the expression of the target in the tumor cells, you need to find an additional target. And BCMA, that it's not mainly, it's not approved for non-Hodgkin lymphoma, express BCMA, and we decided to combine both to increase the possibility of success of treatment of this, avoiding the escape by losing one of the antigen. And this is the situation why we start. We have been able to treat some patients right now. It's clear that there is not important changes in the safety in relation to the use of the different CARs by separated, and we are able to obtain dual CARs in an easy way, and to test this to obtain safety, and probably we expect an increase in efficacy of just CAR19. That it's the comparative element, and maybe in the future we can move for this to other autoimmune diseases where it's more important the role of antibodies and plasma cells at the same time. And the CAR T cells can be one and the other. Both has been able to demonstrate CAR19 and CAR-BCMA some efficacy in autoimmune diseases, but the combination just some centers in China has been using this approach, and we try to make this for maybe in the future or after, to be sure that in this non-Hodgkin lymphoma the safety is clear and there is some increase at least or some similar efficacy in the tumor cells. Later we can move to the autoimmune diseases and autoimmune patients.